All right, thanks everybody for sticking around for one of the last talks on the last day. And thanks to Zach Slagle for giving a really good primer for this talk here. So Zach presented on a lot of the efforts that we did in 2023 in Lake Erie. This talk we've got for you today is really a big overview of the AIS early detection efforts, both in US and in Canadian waters over the last 10 years. So I'll start out with a brief history of how and why the two programs came to be, move into a little bit of what we've done and what we've found over the last 10 years, and then wrap up with some talk about eDNA metabarcoding, which is a tool that will really become more and more prominent with our efforts as we move into the future. So this is a map produced by the Nature Conservancy highlighting areas of high risk for new fish introductions across just US waters of the Great Lakes Basin. The darker cells here are showing areas of higher risk, lighter cells are showing lower risk. The authors of this work really tried to quantify vectors for new fish introductions. So they looked at things like shipping traffic for ballast water releases. They looked at population size for things like aquarium releases. They looked at marinas and the number of boat slips for the potential for bait releases, things like that. So right now you're looking at over 1,800 9x9 kilometer grids across the US waters of the Great Lakes. So to make it a little bit easier to digest, now I've filtered it down to just those grids that are within the top 100. So their relative risk scores are very, very high for new fish introductions. These are the areas that we think are most likely to have a new fish introduced. If you'll notice, Lake Erie does have a disproportionately high risk for new fish introductions in comparison to the other four Great Lakes. 52 of those top 100 grids are in the Lake Erie Basin. Lake Erie, and again, this is just US waters, this doesn't include Canadian data. Lake Erie does have a disproportionately high risk of new fish introductions because all of those vectors that I mentioned are here and they are in full force. So there's heavy shipping traffic, we've got multiple high population centers for those aquarium releases. There's obviously a huge fishery that attracts anglers from all over that could be bringing new species in. So this is, of the Great Lakes, this is one that we're very concerned about a new species being introduced into. So I'm sure a lot of you are aware the updated Great Lakes Water Quality Agreement was signed in 2012. In that agreement, there was a new annex to that directed the US and Canada to establish a binational strategy to address the threat of aquatic invasive species. So directly from this agreement, we had the US Fish and Wildlife Service Early Detection Monitoring Program come online. And then also in Canada, there was the DFO Asian Carp Early Detection Surveillance Program came online. And both of those programs really hit the ground running right in 2013. The whole goal of both of those programs is to detect a newly introduced species very, very early, as soon as possible after it gets introduced, before it becomes established and starts causing problems. The reason we want to do that is so we can maximize the chance of success for any control or even eradication measures, and then also to minimize the cost associated with those control measures. So this here is a map showing over 11,000 sites that we have completed basin-wide over the last 10 years of the program. We have prioritized those locations that have a higher risk for new fish introductions. So that's why you see such a high density of sites in areas like the Detroit River, Maumee Bay, the entire Sandusky area, and then over in the East Basin near Buffalo. As you saw in Zach's presentation, we do a multi-gear approach. We try to sample as many different habitats as we can. The reasoning there being we want to increase our chances of detecting any new non-native species regardless of the niche that it would fill into. Over the last 10 years, we have been able to collaborate quite a bit with our partners. So for instance, in our office around Detroit, we have worked with Michigan DNR and Ontario Ministry of Natural Resources and Forestry on fishery assessments in the Huron-Erie corridor. So that effort helps our partners out with helping to complete fishery assessments that they would already be doing, and it helps us out because we can get some more sites sampled and get our hands on more fish while we're surveilling for new AIS. And I'm pretty sure at the back of the room you can't read that flow chart, but that's totally fine. I don't want you guys to necessarily read it anyway. This is a flow chart just highlighting different decision points when you're structuring an AIS early detection program. So I'm really just going to focus on that last step, which is evaluation and then adaptive management. So given that we're out there trying to find newly introduced species very early on before they're established and causing problems, we're really looking for that needle in the haystack. So in order to do our jobs well, we want to try and be as efficient as we possibly can with our resources. And a big part of that is evaluating our program and then adapting to improve. Typically, the first question we get asked after the field season is over is, did you guys catch anything new? That's a pretty easy one to answer. Typically, that answer is no. Hopefully, that answer is always no. But that usually comes with a follow-up question of, how do you know that if something had been introduced, you would have found it? That one is a little bit more complicated. It raises the problem of how do we gauge our success in finding a new species when we don't know what it is, we don't know where it is, or we don't even know if it's even there. So in our program, we have developed a few different ways to analyze that success and analyze our program. They all really center around one assumption, and that is that there is no totally open niche in Lake Erie that isn't occupied to some extent by species that are already established. So we use currently established species to help us gauge our success for something newly introduced. So very, very basically, the more established species we capture, the higher our chances of finding a newly introduced fish. So this is just a very high-level overview of what we have found over the last 10 years. In 2023, we did pass the 1 million fish mark. We're super excited about that. So we've sampled over 1 million, or captured and identified over 1 million different fish in the Lake Erie Basin across all of the programs, and 117 different species. Over 145,000 of those fish were non-native, comprising 28 species. And then we also wanted to highlight that we did capture over 2,000 threatened and endangered fish, so 20 different T&E species. So we do have a track record of being able to find and identify these very, very rare species that are in the system, which gives us some confidence that we would be able to find newly introduced species while they are still very rare. Looking at our sites sampled and the number of species we've captured annually through time, we have been able to increase our effort and find more species as time has gone on, just ignoring a little road bump that happened in 2020 that I think we're all aware of. But we have recovered well, and again, we have captured more species. That is an increasing trend. So we are learning more about the waters that we work in, becoming more efficient with our time and our resources, and again, increasing our confidence that if something new were introduced, we would be able to find it. So that was some examples of how we've been successful using established species, but I do have some more concrete examples for you as well. This was a Nile tilapia that our Lower Great Lakes team captured in Cleveland while electrofishing back in 2021. We don't really know how it got there, but they did extensively electrofish the area and didn't find any more individuals. So whenever we capture something new, at least on the US side, that information gets run up the food chain to our regional AIS coordinator, and then he goes out and contacts the state manager. So in this case, Travis Hartman was the person that got the call about this tilapia. And then it's the state manager's decision about what, if any, next steps there are. So in this case, since we extensively electrofished it the couple of days after we caught this fish, it is a tropical species, and we were in November already and heading into winter. We were pretty confident that on the chance that there were any more individuals, they would likely not survive the winter. So in this case, we didn't need to take any follow-up action. The next one I've got for you is a red ear sunfish. We detected this in the Black River near Lorraine and in Maumee Bay in 2021. And actually in 2023, we thought we had them in West Harbor Marblehead and in the same exact spot in the Black River. We sent those fish off for genetic analysis just to make very sure that we were right on our ID, and they actually just came back early this week as not being red ear sunfish. So that is a good thing. We have now just those two detections of red ear sunfish still in Maumee Bay and in the Black River. But this is one that is a species that's commonly found in inland ponds in Lake Erie, but up till 2021 we had not detected them in the Lake Erie basin. But those are locations that we're still continuing to go back for, sampling similar habitats in nearby locations just to make sure they don't have an established population and we're not finding any new little pockets of red ear in Lake Erie. The last one I've got for you comes out of the DFO data set. So in June of 2023, the DFO team found a grass carp while electrofishing in the Grand River in Ontario. And then later on that year in August, they had an angler actually report a grass carp capture in Canadian waters of the Niagara River. Both of those fish came back as triploid. But again, these and the red ear and the Nile tilapia are just examples of how we have been able to find new species very, very early on. In the case of these grass carp, those are both locations that we very, very rarely see grass carp, if at all. So we are able to capture these new species before they're really established and causing problems. So last but not least, eDNA metabarcoding. Part of an early detection program and part of your increasing efficiency is to explore new techniques or new technologies that can help you. And this is a very perfect and timely example. So for those of you that aren't as familiar with eDNA metabarcoding, I'm just going to run through this workflow very, very quickly. We start out in the field by sampling water filtering directly from the lake or from the river, wherever we are. All of the material that we filter out gets preserved on this filter, and then we send that filter off to the lab. The lab will extract and amplify the DNA, and then they'll sequence the DNA and assign taxonomic IDs, and then at the end, we come out with a data set of all of the species that were detected on that filter at each site. So that includes both all of the established species that we know to be in the area, plus it's a very good screening tool for any non-native species that we're looking out for, like potentially a tench. So since this is a very new tool, there is a lot we need to learn about it. So in 2023, the U.S. Fish and Wildlife Service conducted a pilot study in Presque Isle Bay in Pennsylvania. There were two main study purposes for this effort. We had a lot of objectives that mainly fell under this broad development of standard sampling methodologies. So we were looking at things like onshore and offshore samples. We were looking at, do we sample at a single point or across a transect? Do we do single filters or triplicate filters? All questions like that. But we also wanted to compare the species detected through eDNA in this effort to the species that were captured through traditional gears over the 7 years that Fish and Wildlife Service had been sampling in Presque Isle Bay. Just for the interest of time, I'm just going to cover a very broad look at that comparison to traditional gears study purpose. But if you're interested, we will absolutely talk to you more about any of the results that we found in this study. So very briefly, we did detect more species through eDNA in Presque Isle Bay in this effort than we have with traditional gears. We got 61 species through eDNA compared to 51 in traditional gears. And at least looking at those accumulation curves, it looked like the eDNA curve was going to asymptote out at a higher level than the traditional gear curve. And I also wanted to point out that the sampling time frame is also fairly lopsided with this study. So the sites sampled with the two methods were fairly similar, 260 in traditional gears compared to 206 in eDNA. But traditional gear sampling took 7 years and 37 crew days, whereas eDNA took 1 year and 9 crew days. So not only is it a very powerful tool from the numbers, we also looked at our results spatially. This now is a map showing where we detected, just one example, tube-nosed goby across Presque Isle Bay using the two methods. So the blue points there are the eDNA detections, and then the orange points are where we captured those physical fish using traditional gears. And there does seem to be a fairly good amount of agreement between where those fish were detected and where those physical fish were captured. This is just one example, but it's pretty consistent across multiple species that we've looked at. So again, this is just another very exciting result that we found from this study. We are working to write up a lot of this analysis and these results to publish here soon. But we do still have a lot to learn about this tool. So the Fish and Wildlife Service is going to do another pilot study in 2024 and likely in more methodology studies in the years to come just to understand the tool a bit better. And both agencies are going to continue exploring eDNA metabarcoding both as an AIS early detection tool as well as just a general fish community sampling tool. So the last thing I wanted to mention is all of our data is publicly available. We have a huge data set, over 1 million fish, that we know is underutilized. So if what we have or what we're doing can help any of you guys out in any way, please don't hesitate to reach out. We do send a lot of our data to the USGS NAS database as well, and we do an annual report every year that we can send to you if you're interested. With that, thank you again for sticking around, and thank you to the Lake Erie community for the opportunity to present here today, and I will take any questions.